Hi everyone, welcome to all. This is Vidyam Academy's most expected question series for medical NEET examination. Today's topic is 12th class biology, chapter number 4, principles of inheritance and variation. And into this subtopic is mutation, genetic disorders. And now it's being presented by Rajiv Patel. Once again, let's welcome to all. Let's get started. The first question is, which of the following correctly defines a mutation in the context of genetics? Option A change in gene expression caused by environmental factors without any change in DNA sequence. Option B, a heritable change in the DNA sequence of organism affecting a single nucleotide point mutation or larger chromosomal segments. Option C, the temporary alteration of a protein structure due to post-transitional modification. Option D, any change in phenotype that is not passed on to offspring. So what is the exact right answer for this question? The right answer is option B. Let's see the detailed explanation for this option. A mutation is a permanent heritable change in the nucleotide sequences of DNA. It's a change in the genetic material itself, not merely in how genes are expressed. Point mutations affect a single base pair, a substitution, one base replaced by another, an insertion, extra base added, or a deletion, base removed. So chromosomal mutations involve larger scale changes. Deletion, duplication, inversion, or translocation of chromosomal segments, or changes in chromosome number. Mutations are heritable because they alter the DNA template used in replication. So daughter cells carry the same change. They can occur spontaneously during DNA replication or be induced by mutagens. So radiation, chemicals. Mutation are the ultimate source of all genetic variation in a population. That's what option B is the right answer for this question. Next, move to question number 2. Down syndrome is a chromosomal disorder caused by which abnormality, and what is the total chromosome number in an affected individual? So here the options are option A, so monosomy of chromosomes 21, total 45 chromosomes. Option B, trisomy of chromosomes 21, total 47 chromosomes. Option C, deletion of portion of chromosome 5, total 46 chromosomes. Option D, translocation of chromosomes 21 to chromosome 14, total 45 chromosomes. So what is the right answer for this question? Yes, the right answer is again option B. Trisomy of chromosomes 21, total 47 chromosomes. Let's go to the detailed explanation. Down syndrome is the most common autosomal aneuploidy in humans, caused by the presence of an extra copy of chromosome 21, making it trisomy 21 instead of the normal 2 copies of chromosome 21. Affected individuals have 3, giving a total chromosomes count of 47. 2n plus 1 is equal to 47. Instead of the normal 46, this extra chromosome results from non-disjunction, the failure of chromosomes 21 homologous to separate properly during meiosis 1, meiosis 2 in one of the parents, usually the mother. Characteristic features include short stature, flat face with oblique palpebral fissure, broad palm with a characteristic crease, intellectuality, disability, so mild to moderate, and increased risk of congenital art defects. The risk of non-disjunction increases significantly with advancing maternal age. That's what option B, trisomy of chromosomes 21. So total 47 chromosomes is the right answer for this question. Question number 3. A male patient has an extra X chromosome, giving him the karyotype 47XXY. Which disorder does this describe, and what are the key characteristics of this? Option A, Turner syndrome, characterized by short stature and ovarian dysgenesis in females. Option B, Klinefelter syndrome, phenotypically male with XXY karyotype, gynecomastia, infertility, reduced and testosterone. Option C. XYY syndrome characterized by tall stature and aggressive behavior. Option D. Down syndrome characterized by intellectual disability and trisomy 21. So, what is the right answer for this question? Yes, the right answer is option B. Let's see why and how. Klinefelter syndrome arises from non-disjunction producing a 47XXY karyotype, one extra X chromosomes in a phenotypically male individual. The Y chromosome is present, so the SRY gene drives testicular development, but the extra X disturbs normal male hormone production and spermatogenesis. So key features are gynecomastia, breast tissue development in males, small testes, hypogonadism, infertility, azoospermia, tall structure, and below average testosterone levels. The condition is not usually detected until puberty when normal male secondary sexual characters fail to develop fully. It's the most common sex chromosome, aneuploid in males with a frequency of approximately 1 in 1000 male births. Let's move to question number 4. Turner syndrome is a sex chromosomal disorder in females. Which karyotype correctly represents Turner syndrome, and what are the characteristics of features? Option A. 45XXO, phenotypically female, short stature, webbed neck and ovarian dysgenesis, infertile. Option B. 47XXX, phenotypically female with extra X tall stature, fertile. Option C. 47XXY, phenotypically male with gynecomastia and infertility. Option D. 47XYY, phenotypically male with above average height. What is the answer? The correct answer is option A. Let's see how and why. Let's go to detailed explanation of this question. Turner syndrome results from monosomy of the sex chromosomes. The affected female has only one X chromosome and no second sex chromosome, giving a karyotype of 45XRXO. 2n-1 is equal to 45. It arises from non-disjunction where the egg or sperm fails to receive a sex chromosome without two functional X chromosomes. The ovaries do not develop properly, so ovarian dysgenesis, a stake ovaries, resulting in absence of secondary sexual characters. So here no menstrual periods and infertility. Characteristic features include short stature, so below 150 centimeter in adults, and webbed neck, and broad chest with widely spaced nipples and lymphedema at birth. So intelligence is typically normal. It is the only viable monosomy in humans and occurs in approximately 1 in 2500 female births. Let's move to question number 5. Sickle cell anemia is Mendelian genetic disorder. Which of the following correctly describes its inheritance and molecular basis? Option A. X-linked recessive affects only males caused by deletion of the anti-beta globin gene. Option B. Autosomal recessive, homozygous recessive, HBS individual are affected caused by a point mutation in the beta globin gene changing, so glutamatic acid to valine at position 6. Option C. Autosomal dominant, one mutant allele is sufficient to cause disease, affects all heterozygotes severely. Option D. Autosomal recessive caused by trisomy of chromosome 11 carrying the hemoglobin gene. What is the right answer? Yes, the right answer is option B. Why and how? Let's check it. Sickle cell anemia is an autosomal recessive disorder. An individual must inherit 2 copies of the mutant allele, HBS and HBS, to show the disease. So here heterozygotes, HBA, HBS are carriers who have the sickle cell trait. They are mostly normal but no partial protection against malaria, which explains the high frequency of this allele in malaria endemic regions. The molecular cause is a single point mutation, a substitution of adenine by thymine at the 6th codon of the beta globin gene, which changes the amino acid at position 6 from glutamic acid hydrophilic charge to valine hydrophobic. This single amino acid change causes HBS molecules to polymerize under low oxygen conditions, deforming the red blood cells into a sickle shape. These sickle cells cannot carry oxygen efficiency, so clog blood vessels and are rapidly destroyed, causing anemia. That's what option B is the right answer here. Question number 6. So phenylketonuria is a Mendelian disorder of amino acid metabolism. Which of the following correctly describe PKU? Option A. Autosomal dominant disorder, the enzyme of phenylketonuria hydrocolase is overproduced, causing excess tyrosine. Option B. Autosomal recessive disorder, deficiency of phenylalanine hydrocolase leads to accumulation of phenylalanine, causing intellectual disability if untreated. Option C. X-linked recessive disorder affects only males, causes abnormal melanin production and albinism. Option D. Autosomal recessive disorder, deficiency of insulin production causing early onset diabetes mellitus. Yes, the right answer is option B again. So phenylketonuria is an autosomal recessive inborn error of metabolism. So both copies of the PAH gene must be mutant for the disease of manifest. The PAH gene encodes phenylalanine hydrocolase, the enzyme that normally converts the amino acid phenylalanine into a trizone in the liver. In PKU patients, this enzyme is absent or non-functional, phenylalanine accumulates to toxic levels in the blood tissues. The excess phenylalanine is converted to phenylketonuria and other metabolites that are exerted in urine. So here critically excess phenylalanine in neurotoxic. It interferes with the normal brain development, causing progressive intellectual disability, seizures and behavioral problems if untreated. Early neonatal screening, GATA test and phenylalanine restricted diet from the birth prevent all neurological damage. That's what option B is the right answer here. Next, move to question number 7. Non-disjunction during meiosis produces aneuploid gametes. If non-disjunction of chromosome 21 occurs during meiosis 1 in a female, what types of eggs are produced? Option A. 2 eggs with 24 chromosomes n plus 1 and 2 eggs with 22 chromosomes n minus 1. Option B. All 4 eggs have abnormal chromosome number 2 with 24 both chromosomes 21s and 2 with 22 no chromosomes 21. Option C. Only 1 egg with an extra chromosome 21, the others are normal. Option D. Non-disjunction in meiosis 1 produces only 1 abnormal egg per primary oocyte. What is the right answer? Yes, the option B is right answer again. Let's see why and how. When non-disjunction occurs during meiosis 1, both copies of the chromosome pairs fail to separate at the first division. In the primary oocyte, homologous chromosomes should separate during meiosis 1. If both chromosomes 21 homologous go to the same secondary oocyte, the secondary oocyte has 24 chromosomes n plus 1 and the first polar body has 22 n minus 1. When these two cells then undergo meiosis 2 normally, both cells divide equally. The secondary oocyte gives 1 egg with 24 and 1 polar body with 24. While the first polar body gives 2 polar bodies both with 22. Its result, all products derived from non-disjunction events are abnormal. 2 cells with 24 n plus 1 and 2 cells with 22 n minus 1. If the 24 chromosome sex is fertilized by a normal n is equal to 23 sperm, the zygote has 47 chromosomes. Trisomy 21, that's what Down syndrome. Next, question number 8. Match column 1 is disorder and column 2 we have here key identifying feature. The first one Down syndrome, second one Turner syndrome, third one Klinefelter syndrome and the fourth one sickle cell anemia. So here A, B, C, D. A is 45, X karyotype, Weber-neck, female infertility. B, autosomal recessive point mutation in beta-globin sickled RBCs. Option C, trisomy 21, 47 chromosomes, flat facial features. Option D, 47 XXY karyotype. So gynosomatia, male infertility. So here the option A, B, C, D. Option A, 1C, 2A, 3D, 4B. B, 1A, 2C, 3B, 4D. Option C, 1C, 2D, 3A, 4B. And option D, 1B, 2A, 3C, 4D. What is the right answer? The right answer is option A, 1C, 2A, 3D, 4B. Let's see the explanation. Down syndrome, one matches C because it's a trisomy 21 giving 47 chromosomes with a flat facial features, oblique palpebral fissures, and intellectual disability. Turner syndrome 2 matches A. The 45X karyotype in females causes short stature, webbed neck and ovarian dysgenesis and infertility. So Klinefelter syndrome 3 matches is the 47XXY karyotype in a male's cases. So gynecosomatia, small testes and infertility with otherwise male phenotype. Sickle cell anemia 4 matches B because it's autosomal recessive caused by point mutation in the beta globin gene. GAG, GTG, glutamic acid, valine, producing abnormally shaped sickle cell or sickled red blood cells under low oxygen condition. So memorizing one unequal identifying feature per disorder is the fastest strategy for match questions. Let's see question number 9. We have assertion A, hemophilia is more common in males than in females because males need only one copy of the recessive allele on their single X chromosome to express the disorder. Reason or hemophilia is an X-linked recessive disorder. Females require two copies of the mutant allele, so one on each X chromosome to be affected, while a single copy on the X chromosome of a male who has no second X to mask it. It's sufficient to cause the disease. Option A, B, C, D are here. A, both A and R are true and R is the correct explanation of A. Option B, both A and R are true but R is not the correct explanation of A. And C, A is true but R is false. And D, A is false. Hemophilia is equally common in males and females. What is the right answer? So here the right answer is option A. Both A and R are true and R is the correct explanation of A. Why and how? Let's see the detailed explanation. Hemophilia is significantly more common in males. The frequency in males is approximately 1 in 5,000 to 10,000, while affected females are extremely rare because they need two mutant X alleles simultaneously. Reason R is also true because gives the precise genetic explanation. Hemophilia A is caused by a mutation in the F8 gene on the X chromosome. Males are hemizygous. They have only one X chromosome XY, so one recessive mutant allele is sufficient to cause the disease because there is no second X to carry the dominant normal allele. So females have two chromosomes XX, a carrier female is phenotypically, so normally because one normal allele produces enough clotting factor 8 to prevent bleeding. So only homozygous recessive females are affected. R gives the exact molecular and chromosomal mechanism that directly explains why males express the disorder more frequently making. That's what. So here assertion and reason both are correct. And final question of this topic, question number 10, arrange the following molecular and clinical events of sickle cell anemia in correct order. P. Thymine substitute for adenine at the 6th codon of the beta globin gene, point mutation. Q. The codon changes from GAG, so glutamic acid, to GTZ, valine. R. Abnormal HBS protein is produced. Valine replaces glutamic acid at position of 6 of the beta chain. S. HBS molecule polymerized under low oxygen conditions, forming long fibers. T. Red blood cells deform into a sickle crescent shape. U. Sickle cells claque capillaries and rapidly homolyzed, causing anemia and vascular-oxalative cries. So here are the options A, B, C, D. A, PQRSTU, and B, QPRSTU, C, PRQSTU, and D, RPQSTU. What is the right answer? The right answer is option A, PQRSTU. That sequence is absolutely correct. You know the answer because the pathological cascade begins with the single DNA point mutation where thymine replaces adenine in the 6th codon of the beta globin gene. Q follows as the direct consequences. This base change converts the codon from GAG, so encoding glutamic acid, to GTG, encoding valine, altering the genetic message. R is the protein outcome when the mutant mRNA is translated. Valine is inserted at position 6 of the beta globin chain instead of glutamic acid, producing the abnormal HBS hemoglobin. Yes, describe the biochemical consequences and like normal HBS molecules with the hydrophobic valine exposed on the surface aggregate and polymerize under deoxygenated conditions. So low O2. Forming rigid fibers inside the red blood cells. T follows these intercellular fibers deform the normally binuclear red blood cells into the characteristic rigid sickle shape. U is the clinical outcome. Sickle cells are inflexible, cannot pass through narrow capillaries. Blood flow vaso-occlusive crisis causing pain and organ damage and rapidly destroyed by the spleen resulting the chronological hemolytic anemia. That's what PQRSTU, that sequence is correctly matched regarding sickle cell anemia. So here we explained 10 most important and expected questions for your upcoming NEET examinations. And next, I will come up with another topic which are very important for your NEET examination. Till so far, I would like to conclude this session with very well goodness. Thank you, and thank you so much to all.